heat transfer, nanotribology, bioheat transfer. Since 2007, he has been involved in the production and characterization of heat transfer fluids, popularly known as nanofluids and nanolubricants. His PhD work on graphene-based nanolubricants is expected, or it was already commercialized in Malaysia in 2018. He's currently working at Research Center for Nanomaterials and Energy Technology at Sunway University, where he designs and develops nanolubricants and nanofluids for automotive and renewable energy systems. Dr. Rashid is also involved in industrial consultancy, corporate social responsibility projects, tertiary and higher education consulting, etc. He feels very honored to have introduced the nanofluids and graphenes by Professor Sarit Kumar Das at Indian Institute of Technology, Madras, during 2007 to 2009. So he's going to tell us about COVID-19, and his background is on nano fluids. He's going to talk to us about augmented thermal conductivity using nanoparticles in polymerase chain reaction for the rapid detection of COVID-19. Dr. Abdul Khalid, the floor is yours. Thank you.
All right, thank you so much, Dr. Krishnan. Good morning to fellow colleagues in India, and hello everyone in the rest of the world. It's my pleasure once again. I would like to thank Professor Nirmala for the invitation. Thank you, VIT. So as the topic was described, I'm going to speak about how nanoparticles can improve one of the most known molecular diagnosis tool in during this pandemic, called PCR. Before I proceed, I would like to make a disclaimer. I don't consider myself an expert. However, I'm an early stage researcher. So having said that, I'm currently based in Malaysia, Xiamen University, which is a branch university of Xiamen University in China. This is how our campus looks like, and I welcome all of you to visit us once the lockdown is over. I'm going to touch upon three key aspects. Firstly, I'm going to give you a background on PCR. Then I'm going to touch upon some results using carbon, metal oxide, and hybrid nanomaterials, which have been used, I think, almost a decade by myself and my collaborators in India and other places. And I'll try to conclude my talk with some key takeaways for the listeners. So let's try to understand what's a DNA. DNA, as most of us would know, it's a deoxyribonucleic acid, which is very, very essential, right? So the human genome has approximately 3 billion base pairs of DNA arranged in 46 chromosomes. What do? Is the information carried by DNA is held in the sequence of pieces of DNA called genes. They are very, very fundamental unit of human, animal, and plant bodies. The question is, why should we amplify DNA? It is in order to study and detect individual genes, or, for example, specific DNA regions or mutations of interest. It is often necessary to obtain a large quantity of nucleic acid for study. For example, what we do is we come across, say, for example, soil samples. What we could do is, in order to figure out whether the amplifier resistant genes are existing in the soil or not, we can determine using PCR. Likewise, one of the most familiar examples that you all would be knowing is to detect or to diagnose if somebody has a COVID, which is nothing but we are going to figure out if the traces of a particular type of virus exist or not in any sample that is of your choice. So the daily COVID-19 tests per 1,000 people as of January 2021 is displayed on the screen right now. You can see our country, India, tops the list. And of course, around the world, the numbers exceed several million a day. Before we go deeper, let me also give you a definition of what is PCR. PCR is also sometimes called as molecular phototyping. So why do we call it as photocopying is because with one single strand of DNA, we can't really determine whether no matter what the type of diagnosis is, we need multiple millions and millions of copies of a particular target DNA in order to determine whatever objective we have. So what scientists and the laboratories do is they multiply a single target DNA into millions of copies. So that's the reason we call it as molecular photocopying. The PCR is a fast and inexpensive technique, relatively inexpensive. I wouldn't say it's totally inexpensive. I'm sure you know what's the cost of each PCR test, right? Is used to amplify a copy and small segments of DNA. So where is PCR used? Already you know the usage of PCR in hospitals, but also you might want to know that PCR is an extremely important tool in forensic investigation. So for example, if a thief leaves behind some traces, we can determine whose, for example, trace it belongs to using PCR. So for example, if a single hair strand is found, what the forensic investigation agencies do is they pick it up and they extract a DNA and they try to amplify using the technique called PCR. Not only here, but also PCR has hundreds of usage in different fields. For example, in genetics, in the study of evolution, in chemistry of life, and so on and so forth. Another important reason why we really have to pay attention to improving PCR or furthering advancing PCR technique is because of the economic significance it carries. So much so that it is expected that the compound annual growth rate is going to exceed 10%. So which means as of now, the market for PCR is about 5 billion, and it is expected to grow by almost 6-7 billions by 2025. So you can imagine any improvement you make is going to have a significant economic value as well. Right. So what do people do in PCR? I'm going to give some background as to, I believe some of you are not from PCR background. Like myself, I'm a mechanical engineer from mechanical background, and I'm working in this field because of the interdisciplinary approach we have. So we have, for example, the sample DNA, which needs to be amplified. It will be combined with primers called forward and reverse primers with some nucleotides, and of course with Taq polymerase. We mix all of them together, and then we go through a different cycles. So PCR is basically based on 3 major cycles. So we have denaturation, whereby a single stranded DNA will get separated into 2 separate strands. We call it as denaturation step. This step happens at about 100 degrees, 90 plus degrees actually. So 90 to 95, 95, 96, and so on. So on an average, people use 94 degrees. Then in the second step, what happens is once the DNA is denatured into 2 single strands, then the second step is called annealing. So in the annealing step, whatever components you saw here, especially the primers, would get attached to 2 single strands of DNA. So when that happens, normally this step happens in about 55 to 65 degree Celsius. And then again, the solution will be heated at up to 60 plus or 70 degrees in order for the extension step, whereby again a double structure, double helical DNA strand would be formed. So basically the heating, it starts with very high temperature heating, about 94 degrees, then it is cooled, and again, the temperature is raised to about 70 degrees. So basically PCR, from engineering point of view, from mechanical engineering point of view or physics point of view, it's heating cooling cycles. So this heating cooling cycle, a single cycle having 3 steps, needs to be repeated for almost 40 times, sometimes 35 and so on, right? However, it doesn't come with problems. So the key problems with PCR is, number one, it is time consuming. On an average, from the scratch, from the preparation of the sample, from the determination of, say, for example, the optimal temperatures for a particular type of sample you're using, and so on an average, it can take from 3 to 5 hours. And if you're doing it for the first time with almost new samples, new reagents and everything, the preparatory steps would even take 10 hours. So we are talking about an extremely important tool, having a very, very high significance in current world, and it is taking too much time. And approximately 40 heating cooling cycles needs to be done. Second issue is the energy intensive, it is an energy intensive process. So for example, imagine in India, I believe the power situation is better now, but in the past, you know how the power fluctuation is, and also there is a lot of shutdowns, right? And also, for example, in other countries where the power stability is not very good, the power becomes a very, very important asset for a smooth operation of PCR-based diagnosis. Third is poor yield. Whatever we do is to amplify a single target DNA into millions of copies. If the yield, which needs to be in millions of copies, is not yielded, then the whole purpose is defeated. So apart from these three problems, there are also other problems such as risk of reaction failure due to contamination, because PCR is an extremely sensitive reaction, extremely sensitive. Even a small trace of foreign DNA can contaminate. Even some minute, the impurities can spoil the whole reaction. And also you will find something here, non-specific bands, and also lack of specificity. Sometimes the bands will not be very specific, creating lots of doubts. And finally, it is expensive, as you already know. So the idea is, when we heat any liquid, the heat propagates from the walls until the center. And the transfer of heat from the walls to the center, the propagation of heat or the transfer of heat solely depends on the very fundamental formula we study in heat transfer, Fourier's law of heat transfer, whereby the K is the thermal conductivity, which determines what would be the rate of heat transfer. This is fundamental. The first year, second year students in engineering, they study it. So the idea is 100 years old. Before 100 years, Maxwell came up with the idea that why can't we combine solids which have higher conductivity, such as metals and so on, and combine it with liquids which have relatively lower conductivity. This is, I think, very well established. For the layman's understanding, this is how it can be put up. For example, you have a complete water stream whereby someone has to cross. So the time it takes to cross through will be higher than if somebody has to cross, for example, land water sequence. So this is on the basis of this idea, what we do is we combine higher conducting materials. For example, as you can see here, the conductivity of silver, copper, for example, carbon is much higher than liquids. So when we combine them, what happens is, for example, graphene has very, very high conductivity. We have carbon-based other materials, CNT, multi-wall CNT, and so on and so forth. When we combine such materials with, for example, water, sorry, the value is wrong. Water has 0.6 watts per meter Kelvin. So you can imagine 0.6 watts per meter Kelvin combined with such a high conductivity, the combined effect or combined effective thermal conductivity, which will be much, much higher. So that's the basic idea behind why we incorporate materials into a reaction whereby the heating cooling will absolutely depend on the conductivity. So we follow this methodology whereby we do material synthesis followed by characterization, and then we ensure that the fluids is stable and so on and so forth. Ultimately, we do some PCR experimentation to determine whether the particles are indeed enhancing or not. So here are some examples of materials we used in the past. So we have used, for example, hybrid nanomaterials, which are a combination of 2D, 3D structures. We have done a lot of modification surface. We have done a lot of hybridization and so on. We also developed some 2D structures, as you can see here. And these are metal oxides, alumina, copper oxide, silver oxide, and so on and so forth. So the very first publication in this field came about in 2010 when I was at IIT Madras. So in this result, what you see here is with an optimal concentration of nanoparticle, you can amplify or enhance the yield of PCR. So this is a positive control without any nanomaterials incorporated. So what you see subsequently, when you keep increasing the concentration of nanoparticle, the yield increases. But of course, as the concentration of the nanoparticles are increased, we are also we are required to take note of the specific heat, as the specific heat is going to determine the fidelity of reaction. The more you add nanoparticles, the specific heat is going to increase. That will affect the fidelity of PCR itself. So the optimization process is extremely critical, and we determine that. Secondly, some people argue, nanoparticles do not necessarily have an effect. Rather, it could be, for example, the effect of conductivity of nanoparticle may not necessarily have a direct effect. Rather, it could be some other mechanism such as DNA binding along the nanomaterial, or the polymerase, getting in contact with the material, and as a result, augmenting the PCR and so on and so forth. So to defeat those kind of arguments, what we did is we took out the supernatant of the nanoparticles, and then in order to see if there are any chemical reactions and so on, we did a PCR with only the supernatant of the nanoparticle. In that, what we found is there is literally no impact of the particles chemistry on the PCR reaction. Then we used graphene. This was using TiO2 metal oxide. This is a graphene based. So similarly here, what we found is a very nice bell-shaped curve. You can see with lower concentration, you get lower yield. With as the concentration goes up, the yield, the PCR yield increases, and when you keep continuing or increasing the concentration, the yield decreases. This clearly indicates that the effect of concentration of nanoparticle is extremely evident, contrary to all the other mechanisms that are proposed by the biologists, right? So the similar experiments or similar results can be found in those papers, whereby there are actually hundreds and thousands of papers on nanofluids, where it has been established as you increase the concentration of nanoparticles in fluid, the conductivity will increase only until a particular point. Further increase in concentration will lead to sedimentation, and as a result, the conductivity will go down. So this can be corroborated very clearly in this result, and this paper was published in 2012. Then we proposed mechanism as to why the enhanced conductivity effect will be more prominent, or could be more prominent than any other biomechanisms that are produced by our counterparts in the biofield. What we say is, when you say, for example, have 2D structures such as what you see on the screen, the conductivity mechanism is clearly established. For example, what you see is when 2 or 3 different flakes get in contact in a volume, reaction volume, the effect of percolation effect will become prominent, and the heat transfer path will become more shortened, and the heat transfer rate will also be augmented, increased. So this has been already established by many people around the world, and we use similar mechanism to justify why we believe the enhanced conductivity effect of nanoparticles will be more prominent in case of PCR as well. Then we used, for example, acanthamoeba. This was quite a recent study in 2019, if I'm not mistaken. We used hexagonal boron nitride, which is again a 2D material. So we used similar material because it has a different, it comes from a different family of materials. Nevertheless, we wanted to establish if this can help as well. The reason being, TiO2 and graphene are relatively expensive materials compared to hexagonal boron nitride. Even though nowadays graphene is being produced at much cheaper price, 2 years back it was still regarded as an expensive material. So we took up HPN, we synthesized, and then we determine again HPN can also give you enhancement, but the fold of enhancement may not be as good as the graphene or other high conducting materials. And we again came up with a mechanism which is explained in this paper. You can refer to this later on. One of the key issues with PCR is the duration, the time it takes. So first thing is the yield itself. We have already established clearly that the PCR yield can be improved significantly, almost 10 folds or even more. Now, what we are showing, what you see on the screen is not only you can enhance the yield of PCR, the DNA can be yielded much higher in quantity, but also you can reduce the temperature at which, for example, from the 3 PCR steps, the denaturation, annealing, and extension, which happens at a particular range of temperature, you can bring it down. So what it shows is when you have nanoparticles incorporated in the liquid, the specific heat capacity of the liquid will go up, and as a result, you don't need to heat the, for example, the PCR reagent at 94 degrees, which was the case without the nanoparticles. Now you can achieve denaturation, for example, at 90 degrees or 89 degrees or even 88 degrees. So this was established very clearly. We have displayed all the readings here. You can refer to this paper to see all the reductions that we achieved. So first is the yield enhanced. Second is the reduction in cycle, the step temperature. The third one, what we found is you can also reduce, for example, the cycles, number of cycles that is required in PCR. Normally, to amplify a single target DNA into millions of copies, there are approximately 40 cycles. What we found is you can reduce the number of cycles so much so that it could be as less as 20 cycles. Within 20 cycles, you can clearly determine whether there is amplification or not, whether the, for example, for PCR diagnosis in case of COVID, you can clearly establish whether there is the result is positive or not with the reduction of 20 cycles. So I think this will cut down almost 50% of the reaction time, and this has been also established in one of our studies. Okay, so what do you see here is the effect of hybrid. So we have established the PCR efficiency could be improved using graphene, carbon-based materials, hexagonal boron nitride, metal oxides. Different metal oxides have been used: copper, silver, alumina, and so on and so forth. We also established using hybrid materials. So if, say, for example, you have a 2D structure, you can decorate it with 3D structures, and with different combinations of 3D structures, such as what you see on the screen is graphene oxide, TiO2, and Ag silver can form a fantastic hybrid material with a better, higher conductivity. But the only challenge in this case is the stability. These particles are highly dense, and as a result, it will tend to settle down. Nevertheless, you can still see enhancement taking place. This also proves that many materials which are chemically not going to react with the PCR reagents can be formed as hybrids, and you can use them as potential PCR enhancers. Finally, I have a few more slides, two or three more slides, and then I'll try to wrap up and would be happy to take any questions. So what you see also is a nanoparticle interaction with DNA sequencing. So in our recent study, we established that there is indeed some binding of nanoparticle and the DNA. However, it is not as strong enough to pull it from passing through the gel pores, the agarose gel pores, in order for you to determine the band intensity using gel electrophoresis. Then we also determine the DNA sequencing, which shows that the mutation to DNA, which is used as a target, is not really severe. Rather, it is almost neutral. So meaning using nanoparticles, you are not really damaging the DNA. And also what we determined or established to solidify our argument that thermal conductivity is one of the most significant mechanisms behind the enhancement of PCR by including all the different types of nanomaterials. What we do is we use a very simple UV-Vis technique. What you see here is by heating the sample, whereby you have DNA and different nanomaterials, we establish that denaturation can be much amplified in case of nanoparticle present samples, and you can see the band intensity or the absorbance showing that the presence of single stranded DNA are much higher in case of different nanomaterials we have selected. And also the denaturation can also happen at a much reduced temperature. This could also be established not necessarily using PCR, but also using UV-Vis technique. So we used different concentrations and found similar results. We didn't stop there. We further extended our studies to CFD, computational fluid dynamics. We did establish that using nanomaterials, you can achieve much higher heat transfer rate in all the three steps for different nanomaterials. This is for thermal titanium oxide nanoparticle, and this is for hybrid nanoparticles. So in conclusion, I would like to say that the enhanced conductivity has largely been ignored by the biologists. If you take nano PCR papers from around the world, I think our group is the only group which vouches for enhanced thermal conductivity effect. And we have come up with many mechanisms. We have proposed it. Nevertheless, there is still scope for significant, improvement, and much more can be done. Number two is interdisciplinary approach to underlying mechanisms behind nano PCR enhancement will probably pave way for more inclusion and modification of many nanoparticles. What you have seen, the results I shared is from probably 6 or 7 papers. There is much more could be done with many hundred nanoparticles that exist, and also many hundred combinations of different nanoparticles that we have. Probably Professor Nirmala can do wonderful combinations of nanomaterials, and with which we can study numerous PCR studies, carry-out PCR studies. Number three is not many studies are using nanoparticles in RT-PCR. What we use for coronavirus diagnosis is the RT-PCR. The results that I displayed is conventional PCR. So not many studies have been done. If anybody is interested, I'll be more than happy to collaborate, and we can extend the study further. Fourth conclusion I would like to mention here is augmented thermal conductivity using nanoparticles in PCR for rapid detection of COVID-19 is absolutely possible because of all the results I have just presented. For the final conclusion I would like to mention is the future is promising for nano, 2D, and hybrid materials for the COVID-19 detection or any potential future virus detection using PCR. I would like to thank my professor, my mentor, Professor Saeed Kumar Das, who introduced this topic in 2008. And since then, I'm working on this field. And also, I would like to acknowledge all my colleagues in all these different universities, my mentors, my supervisors, my students, and also collaborators. Thank you so much. If you have any questions, I'm more than happy to take it now.
Thank you for a wonderful talk, and it's very important. And in molecular diagnosis, still it is emerging. Although there's a lot of advancement in the field, the sensitivity of detection, and then the questions of false positives and false negatives still looms large in the PCR-based diagnostics. So I don't see any questions from the audience. And okay, as a biologist myself, I would like to, you briefly touched on RT-PCR, which is very important. So reverse transcription and PCR, because to all those audience, PCR is based on from the DNA. But if you look at many of the viruses, they have RNA as the genome. Okay, so we cannot do PCR directly with the RNA. We need to make it as DNA. That's a process known as reverse transcription. Then use that template for making PCR. So that is going to be very important if we really need to make a breakthrough, and especially using these kinds of nano fluids. The important thing is that all along, you described wonderfully about use of nanofluids in your PCR-based assays. So you've just done it with agarose gel electrophoresis, but it is important that you do a real-time PCR, because that is where we use actually fluorescent probes and then monitor them. And that is more quantitative, because See, the thing is that with gel electrophoresis, what happens is that it is based on visualizing under UV light the quantity of DNA, which is not completely quantitative. After some time, this saturates, and you wouldn't be able to actually exactly quantify. Okay, so if at all, if you really wanted to take it to clinical settings, this is just a comment. You need to think in terms of doing it with real-time PCRs. So you don't have to run the gel. So they have fluorescent probes, like for instance, cyber green. A number of other probes have come out now, and efficient probes have come out, and then they quantify DNA based on these probes. Only thing is that you need to see whether the quenching of any of these dyes happen in presence of your material that you study. And invariably, the enzymes we use, magnesium chloride or magnesium sulfate, they're required for enzymatic activity. I see that there's no disturbance in the activity, but in case if it is done in the future, these are all comments I'm giving you. So you need to consider whether these metal ions like magnesium would interact with any of your nanofluids. So that is a key. So otherwise, it's a very exciting field, I feel, and a lot more can be done. And if we can, reduce the number of false positives with this technology, then the accurate diagnosis can be really, really helpful, because as on now, COVID-19, the detections we hear, at times it shows positive, at times it shows negative. So these are some of the limitations that we have. We are living with it right now, but whether it can be eliminated totally. So that's a comment. Basically, it's a comment. It's a wonderful talk, and thank you so much. So I'll just see if any more questions are there.
Thank you, Dr. Krishna. Really appreciate your comment. Yes, you're absolutely right. This is not the technique for quantitative diagnosis. Quantification is definitely not possible in this one. But like I said, these are all preliminary studies.
Sure.
And we definitely need to take this forward. And the key for further studies should be based on the enhanced thermal conductivity effect of nanoparticles, along with all the other biomechanisms that may be possible. We are not completely ruling out.
Sure.
But what we are trying to suggest is the key mechanism that cannot be ruled out whatsoever is the enhanced thermal conductivity effect of nanoparticles.
Definitely.
So that's the key message I'm presenting.
For sure. I agree with you. Totally agree with you. So there is one of the audience has asked a question: how to identify the material to use biological application? This is a very broad question. So.
Okay, I mean, in the context of PCR, to whoever this question asked, the way we determine is, normally if you go to any molecular biology lab, on a given day they run hundreds of PCR reactions. So you could, for example, if you're very good in synthesizing nanoparticles, you can come up with different combinations, and you can based on the literature, you can choose to work with a particular concentration. And then include it in the PCR reaction and see if the reaction is, in any way inhibited or not. If it is not inhibited, that gives an indication that this particular material you're using might be a potential enhancer. Then what you need to do is carry out, the gradient PCR with different temperatures and so on and so forth, and also with different concentration to establish what would be the ideal concentration. And perhaps the next step would be, like Professor Krishnan mentioned, you need to take it for, RT-PCR and perhaps be more accurate in your quantification. I hope that answers your question.
Thank you. Thank you. So there are no more questions?
Krishnan sir.
Yeah, ma'am, please.
Dr. Rashid, it was a very good presentation. Thank you so much. I just have one question. You were telling about because of the increased thermal conductivity of the materials which are used in the PCR reaction, that is your nanofluids, you can reduce the number of cycles and also increase the yield and so on. So, and it is also feasible to apply the same concept with RT-PCR. which is currently used for COVID detection. So what is your expectation? How much can you reduce the time? Suppose if it is 4 to 9 hours is the current time required for detection. So how much time reduction can you expect by following this method?
All right, thank you, Professor, for the wonderful question. All our reports are based on conventional PCR. So I can give you my perspective on conventional PCR and the achievement we have got by reducing the time and cycles and so on. So in terms of, for example, temperature, right? As I described during the talk, we managed to reduce the temperature by several degrees. I would say in case of denaturation, we managed to reduce until 89 degrees. So denaturation could be done at that. Secondly, we also reduced the step time. You know, each step has a particular time. So we managed to reduce the time by 10%, 20, 30, 40 until 50%. Yet we could see the amplification happening. Number three, in terms of cycles, you know, you can gradually decrease the number of cycles from 40 to 35, 30, 25. So we managed to detect, we managed to see the yield until 25 degrees. In case of positive control, there was visibly no yield at all. But in case of, with the use of nanoparticles, we could see yield happening even at 25 cycles or even 20 cycles in some cases. So, so when you are reducing half the number of cycles, so you're actually talking about reduction of 50% time, I think, which is really humongous and huge. Nevertheless, when I said 8 to 10 hours, it is taking into account all the preparatory steps, which may not necessarily be repeated for every single time. So preparatory steps are only done once you have new reagents and so on, once you need to establish the optimization. So I hope this answers your question.
Yeah, it's very nice. Yeah, it will be very important because in a clinical setting, the robustness with which we handle number of samples per day per productive time, that is going to matter a lot. And also, if you reduce the number of cycles, okay, so instead of doing 35 cycles, 40 cycles, if you do it at 20 cycles, 25 cycles, it will also reduce the cost of the materials. For instance, the fluorescent probes, they're extremely expensive. You can cut down the cost of the PCR, analysis. If you can just, the cost, if it can reduce, instead of paying 2,000 rupees per test, if one can get it done at 1,000 rupees or 1,200 rupees, it's a lot of cost saving method, I would say.
Yeah, thank you, Dr. Krishna. That's a very important point, which I missed during my talk. Actually, yes, what we have also done in some of the studies is we reduce the quantity of Taq polymerase. I think that's one of the expensive materials in PCR. So we managed to reduce different quantities, different reagents in various quantities and saw the impact. So what happens is, yes, in the presence of nanoparticles, you can also play around with the concentration of PCR reagents, yet achieving similar results. So yes, it also helps you economically. Yeah, thank you so much.
So there's one last question. So what are the challenges one can anticipate when you do an RT-PCR? So this is one of the audiences has asked the question.
Okay, for your information, like I said in the talk, we haven't really done any major study using RT-PCR. However, we have a current collaboration with the University College London, UCL, the I think virus department. I don't know, not very much familiar with the bio terminology. So we have a collaboration. Currently, it's ongoing. The major challenge we are finding is the optimization itself. So you work around a particular concentration or a series of concentrations, which give you good yield in conventional PCR. But in the RT-PCR, that may not be the case because you work with a different mechanism. RT-PCR works on fluorescent basis, which is not the case in conventional PCR. So we did find some challenges when it comes to that. Nevertheless, it's an ongoing study. Would be happy to share those results in future. Maybe you can follow our work and look for the publication when it is out.
Yeah, just adding a point. So if you take RNA or DNA, DNA is more stable. Okay, it readily forms double helix and very stable. RNA is very unstable. Usually it is single stranded. Okay, RNA double stranded is there, but it is not so much, not prevalent so much. And RNA is highly unstable and it gets degraded very fast. And everywhere in nature, there's a lot of RNA degrading enzymes are present in our hands, external bodies protected by these RNAs. So you have to handle RNA very carefully. So that's a major thing. And also the enzyme that transcribes, reverse transcribe, they are also highly sensitive. So this is where the problem could come. Okay. All right. Thank you, Dr. Abdul Khalik Rashid, for a wonderful talk. And you put everything in a simple perspective in a layman's language. Thank you so much. On behalf of the organizers and from VIT, we would like to thank you for giving a wonderful lecture. Over to Dr. Nirmala Grace, ma'am. Thank you.
So, yeah, thank you, Dr. Krishnan. Once again, thank you, Dr. Abdul, for this nice talk. And thank you, Dr. Krishnan, for chairing this session. Thank you very much.
Appreciate it, everyone. Thanks for the invitation,
Thank
Nirmala.
Thank you.
Yeah.
Have a good day.
Yeah.
Bye.
Good day. Bye. Yeah. Participants will join back at 11:15 a.m. for the next talk.